Our speaker this afternoon is Dogic, or actually somebody told me it's supposed to be Dogic.
No.
No?
Jogic.
Jogic. Okay, Jogic.
No, but I'm American. For better or worse, I'm Dogic now. So, you know.
And he's from Brandeis University. He's an experimentalist and has done a lot of work on liquid crystals in the past, colloids, but most recently on a beautiful example of active liquid crystals, especially in mathematics, and that's perhaps what the talent about. We'll see.
Okay. Yeah. So it's not clear to me actually what I should lecture about. So I was thinking, well, maybe I should email Christina and Vincenzo and ask them what I should lecture about, but then I was afraid that Tina might say, you know, you should derive the hydrodynamic equations of active nematic liquid crystals and show how they're related to your experiments. And then I would actually have to learn this stuff. So, so I was postponing it, and then suddenly it was like this weekend. And it's a blue screen. So then it was a few days ago, and so then in panic state I emailed all the people who I thought did interesting contributions to active liquid crystals, and they sent me PowerPoint slides. And then I had to study them right over the weekend and understand what they mean. And so last night I was up until 2:00 in the morning sitting in my dorm room. So I really felt like an undergraduate again. It was, okay, the other thing that I want to say is my handwriting is really bad. It's comparable to John Toner.
That's a strong statement.
We can have a competition. Okay, well, but that was funny, right? Because John scanned his bad handwriting in a computer. That really didn't make sense, right?
Are you saying that was cheating?
No. I mean, it was. Anyways, I don't want to say anything more. So what I'm hoping to do is I have 3 lectures. I have 4.5 hours to talk about. First, I'm hoping to give you a broad overview of experimental systems that exist of active matter, and what I think are important results, and especially in the first lecture. Then in the second lecture, I'll really focus more on our own system, but then I can put it in the context of all these other experimental systems that have been done before. And then in the third lecture, I guess I hope to show you some data that's I know this is supposed to be pedagogical, but when we are in the lab, we actually don't know what we are doing most of the time. And so, I hope to give you some results that we have, but we don't understand. And so hopefully, that can motivate some of you young people to, spend the next few years thinking about it. Okay? Are there any questions? Yeah, so yeah, it's PowerPoint. I'm sorry about that. Because I got that's the only instruction I got. I got an email saying you should give a chalk talk, which I ignored. All right, so yeah, so this is really going to be an experimentalist talk, right? And I'm talking about things that people in the audience know much more about than I do, because I've literally been reading these papers this weekend. So maybe they will help me out. Yeah, so equilibrium thermodynamics, right? So, in general, if you have a low temperature, you will have in this XY model, you'll have all the spins pointing in one direction. They'll form a quasi-long range order, and so that's favored by energy. And then if you increase the temperature, you'll undergo phase transition where entropy dominates, and you have a disordered state. And and so there's a general prescription for how to calculate these things, right? Is you write down the free energy expression, and you write it down, and I guess you've heard a lot about this. You can write down this free energy expression really by considering the symmetries of the system. And that's how I'll try to classify also the non-equilibrium systems that I'll talk about according to not, their microscopic particulars, but what are kind of the general symmetry properties of the active matter systems and what I think are kind of interesting experimental realizations of them, right? And so you minimize, but so in equilibrium, there's a, well-known prescription that how you minimize the free energy that determines the equilibrium phase, right? And so what we want to do is we want to, study these kind of systems out of equilibrium, right? And so in an equilibrium, again, you can have different flavors of interactions that are going to have different symmetry properties, right? For example, in this spin system, you have polar interactions, right? And that means that two spins want to be pointing in the same direction, right? Instead of a nematic system in which, there's no arrow. And so there's kind of multiple different things that you can the kind of interactions that you can think about. Again, these polar interactions, right? So that's your spin system, and in equilibrium, what you say is that this spin wants to be aligned next to its neighboring spin. And so people have studied these kind of systems to a great extent. Then you can have a different symmetry of interaction, and that's a nematic interaction, right? And here you don't have an arrow, right? So if you have two rods in a nematic, you have an arrow, these molecules are polar, but the interaction between the systems does not depend on the arrow, right? And so these are effectively an arrowless rods, and the order parameters that you use to describe this kind of system are different. And then I guess you have the simplest system is an isotropic interaction. So these are just kind of hard spheres, and so when two of these things interact with one another, there's no effective torques between the particles. Okay, so you can write down, so far this is all equilibrium, but then you can drive the system out of equilibrium with activity, right? And there's different kind of microscopic activities, right? Is you can have, polar activity, and that means I have my, an animal or a spin, and it's moving in one direction, right? And it might move forward noisily, but there's a definite arrow which you can assign to the movement of the particle. And then you can also have nematic dynamics, which is, I would think, more rare, but it does happen in a number of systems, in which molecules move forward for a while, and then they move backwards, right? And so, there's no net displacement in this kind of activity, right? And so you can have particles that can have these kind of interactions, and that can have this kind of activities, and you can now start cooking up different kind of systems and ask what is the behavior that emerges, right? Okay? And so that's what I will try to do, right, as I go through this talk. And then there's obviously the important question is also hydrodynamics, right? Whether these things just crawl on a solid surface, or whether they live in a fluid and they talk to each other by inducing fluid flows, right? And so that will also modify the behavior of the system. And so, as an experimentalist, you want to come up with simple systems, right? And that's challenging simple active systems, right? And so, there's all these very, very simple theoretical models that people propose, and they say, that will describe behavior of this very complicated, herds of animals and things like that. But and there's actually people who do experiments on flocks of animals and things like that. But as a I spend my graduate studies measuring phase diagrams and doing quantitative tunable experiments. It's not so pleasing to a physicist, I guess, right? And so you have this complexity, right? And so you actually, I guess one could argue that you have many examples of very complex systems of active materials. But then as you move towards less and less complex systems, things become far few and between. And by that, what I mean is you want to have a system in which you can vary all your microscopic parameters to really rigorously compare it to the experiments. Okay? And so that's challenging, right? Because if you're studying equilibrium liquid phase separation, you can literally, just take your cup of water and that's your experimental model system. But if you want to study model systems of active materials, you really have to, put some work into it where you can get something that's tunable and controllable and that might tell you something that might be relevant for experiments. Okay? So I would say that there is, in general, 3 different categories of systems, right? And they all have their own advantages and disadvantages. And as I go through the systems, hopefully I can tell you, what from my own personal perspective, what are advantages and disadvantages of each, right? So you have synthetic active swimmers, and I'll talk about these a little bit. So these are literally colloids. Christina showed you some movies that swim around. And so the advantages of this system is that they're very tunable, right? And so you really engineer the system from the bottom up. And and you can control the microscopic dynamics, as I'll show you. There's the disadvantage is that there's very few microscopic mechanisms that can power propulsions of particles. There's essentially only one, and I'll talk about that in some detail, right? And so we really need clever people to come up with clever mechanisms that will propel dynamics of microscopic objects. But that's really challenging, and it's really more a thing. You need to be a chemist, right? You need to somehow engineer you need to engineer catalysis and couple it to large-scale conformational changes. And I actually have I don't know how I would design one, right? The other disadvantage of synthetic active swimmers is that they are orders of magnitude less efficient than biological swimmers. I saw one paper where it says that one of these catalytic swimmers is 9 orders of magnitude less efficient than something like a motor that powers a biological organism, right? And so this creates problems because it's very difficult to achieve steady states. That the system might take a while to settle in a steady state.
So what exactly do you mean by efficiency?
There's many different ways to define efficiency, right? So I have to go back to So I mean, what this particle hydrolyzes hydrogen peroxide. I'll talk more about it. And you can calculate the energy release associated with this motion, and then you can compare it to the efficiency that you get out, right? And you can also do that for a molecular motor. It hydrolyzes ATP. You know how many KTs are in there in one, in ATP, and you can compare it to the work you can get out, right? But things get, I'm not an expert on this, right? So I can show you the papers where one could look. The other option is, okay, well, we just take living organisms, right? And so living organisms, by definition, are robust. I mean, they're in a certain way they are robust, right? They are out there, in the nature trying to survive every day. And so they've been optimized for efficiency, unlike your catalytic colloids that only exist in a lab environment. So if you have a bacteria, it can live for many hours or days, right? But so the drawback of living organisms is that it's very difficult to tune its microscopic activity, shape, and interactions, right? So it's very so these things are not easily tunable. If I have a bacteria, it's actually very challenging to make it go faster, to make it go slower, or things like that. Although people are making are making progresses in this area, right? And the other thing is that living organisms, by definition, are living because they reproduce and die, right? And so to tell a living organism that you don't want it to reproduce is also very challenging, right? And so in these systems, you usually don't have the number conservation, which complicates the equations that John talked about. Okay. Oh, well, okay, so simplifies.
I discovered years later that it's actually simpler with this reproduction to get.
Okay, because there's, maybe he'll.
For technical reasons that
Okay.
I'm happy to tell anybody about in private.
Okay.
Turns out to be true.
All right, so maybe my worldview says, I have to change my model system, right? I can't make microtubules reproduce, though. Actually, you could have them nucleate, and so people do, and that actually is important in some biologically relevant things. So, yes. So it's very important. So, okay, and so, but so this makes it very difficult if you have a batch of bacteria to really make it, be in a steady state for days or that you would want, right? And so the third system, and that's what we do in my lab is we develop biomimetic systems, right? And so we take parts of living organisms that are very simple and tunable, and we try to put together organisms. We try to put together these building blocks into some higher order assembly that might exhibit novel dynamics, right? And I think that's interesting because living organisms by themselves, they are tightly constrained, right? And so if you put non-equilibrium pieces together, you can build a lot of different kind of functionalities and structures, right? But a lot of these things are not viable out there in nature, right? Because an organism that would use this kind of motility mechanism would never have a chance of surviving, right? And so by building things from the ground up in the laboratory, you have a chance to explore things that are far from equilibrium, that have no biological relevance, but are interesting never to say, right? And I hope to show you some, especially as we go towards the end of my talk. But there's no there's no ideal system, experimental system of active matter. Each one has its advantages and disadvantages, and so you have to kind of carefully choose what you want to, which system you want to work on and what kind of question you want to address. Okay, so this is what I hope to cover today. I'll tell a little bit, and this was certainly done by Christina already earlier today, about the simplest system that you can imagine: isotropic interactions and polar active dynamics. Then I'll talk about polar interactions and polar active dynamics, and this falls into the category of Vishek model. And then I'll talk about apolar or nematic interactions and nematic active dynamics. And sometimes it's actually not clear which category the systems belong to, okay? And so, so catalytic swimmers. So let me talk about that first. So as far as I know, and this is what I was told, is this is one of the first systems that describe the catalytic swimmer. I don't know if this is correct, actually. In the lab of Whitesides, they made a large PDMS structure like this. It's a millimeter size, and they've just put a little rod, platinum covered rod, into this PDMS, and they put it in a hydrogen peroxide solution, and this thing takes off and surfs on the air-water interface. And that's, when I was putting this talk together, what I actually thought is like a lot of these advantages experimentally are just really pretty simple, right? It's not that complicated, and you just have to have the right idea at the right time, right? And so, this was published in 2002. I wanted to get a movie of it, but that was they just published and they had a figure of this thing swimming around, but there's no movies, right? And so then there was a person at Penn State who saw this, and he said, well, I can do something similar like that. And so what he did is he created a catalytic swimming nanorod. And so now this is this is a microscopic structure, so it's about the size of 1 micron, right? So it's Brownian in addition to being powered. And so it has 2 components. There's the gold and the platinum part, right? And so when you put this in a hydrogen peroxide solution, platinum is going to catalyze hydrolysis. Does hydrogen? Is there any chemists in here? Or I mean, H2O2 breaks down into H2O and O2, right? And so this creates an asymmetric diffusion of ions, and that propels the molecule forward, right? And so this was done by a group of Sen, and this was published in JACS 2006. This paper is amazing because, I mean, I was just reading it this weekend for the first time. Most of it is wrong, right? I mean, I saw this and like, does anybody see something wrong with this graph? This was published in JACS, right? And they can publish stuff without graphs. I don't know, this could be meters or centimeters. You can figure it out because actually this should be 2 microns, and he says that this is 2 microns. So this is very likely microns, but the fact that they and then they have a theoretical explanation that's, I think, is completely wrong. But it's really an important paper, right? So you can like write a paper, be mostly wrong, and still make an important contribution, right? But if a student in my group meeting shows this, right, he has to go out and run around the building for 10 laps, right? No axes on the labels. I mean, anyway, so, huh?
No labels.
No labels. Sorry. But anyway, so you can see these things, right? And these things are taken 5 seconds apart. So I presume that this is one of these rods, right? And you can see, again, I tried to download the movie, but it was in some weird format, and I spent 3 hours trying to decode the codec while I should been preparing the lecture, so I gave up after some point. But you can see this rod, and it's moving around, right? And so this moves, so this is, assuming that this is microns, right? It's from 90, so it moves 20 microns in 5 seconds. So it's moving at 4 microns per second, right? And so, so that was an example of a microscopic swimmer. I think it's one of the first ones. I'm not sure, right? But I think it is. And so that really took off, right? And so I guess there's no chemists here, right? So what I can say is that chemists have been perfectly, as far as I can tell, they've been perfectly happy reproducing these results and literally publishing hundreds of papers that more or less say the same thing. That's what they do, right? Sorry. No, they publish a lot of papers, right? They're just. Oh, shit. Erase. Okay, I'll be more politically correct. Now, chemists are great. They invented all of this, right? We would still be doing Ising model if it was physicists, right? I guess, they could say physicists have been solving Ising model for 50 years, right? I'm sure they have the same. But, the obvious thing to do is you have the swimmer. Why don't you just put a bunch of them together, right? And see what happens. And so that took a long time, right? And honestly, these things are technically challenging. This thing creates oxygen, and it creates bubbles, and there's a lot of challenging things that have to be overcome. So they did a really important thing, but then I think it's important to move, to see what happens as collective behavior, right? And so that really waited until 2013, 7 years later, where again Christina already alluded to this work by Paul Chaikin and Jeremy and others, and so they're really experts in synthesis there. And so what they did is they synthesized this particle. This is actually Stefano, I think. Is you have a micron-sized particle, and it has this little tail. And this is hematite, and this hematite again hydrolyzes hydrogen peroxide when you shine it with blue light. If you turn off the blue light, you have complicated chemistry happening, which I don't understand. Chemists are figuring it out. I don't know how much of it they understand. Is when you shine blue light, this thing is just a passive particle. When you don't have blue light, this thing is just a passive particle. You shine blue light, you induce the catalysis of hydrogen peroxide, and that propels the beads forward. And that's shown in this movie over here, right? Oops. Hopefully. Right, so you have these guys sitting around, light on. It takes a while. You see some of them just hang around and then they suddenly get kicked and they start moving around, right? And so this is in a very dilute suspension, and they're moving in microns per second. This is probably 50 microns or so. Okay? And so, so this is great, and I think the important advance was the light control of the hydrolysis, because before you would just put all of these things together and things would happen so fast that before you had a chance to go under the microscope, you would create bubbles and all kinds of messy things. Okay, and so now you can just simply have your beads, let them sediment, you turn the light on, and you start observing this clustering that Christina showed before, right? Do you think that a chemist would actually listen to my lectures? I don't think. Okay, sorry. So I'm thinking about that I got myself in trouble here. Yes.
So can you control the intensity, like the activity?
Yes, I will get to that, right? So again, I think, so this is reminiscent to what, this is what Christina showed. But I think what happens in this system is that they never coarsen, right? So they reach a certain size and then they stay at that certain size, right? And so the microscopic mechanism that governs the dynamics of these particles, it's much more complicated than what Christina showed before. And I don't know if it's fully understood. Yes?
The particles are active only in they're in contact with the fluid, right?
They are all in the fluid. So this is a two-dimensional chamber.
Yeah, like the one in the middle of a cluster.
Yes.
Is it still active?
That's a question I have, I have also, right? Because then if it's in the middle of the cluster, it really doesn't have a gradient in a chemical, right? And so then it's round, it's still undergoing Brownian diffusion, but I don't know if its activity might change, probably does. But, do you know, Christina?
I don't know. I know they look much more stationary.
Yeah. Yeah, I mean, their propulsion dynamics is certainly much more complicated.
The activity switches on both cell propulsion and sound attraction.
Yes.
And although they kind of show that it is the cell propulsion that is the clustering, that is the attraction that is the effect that is clustering and not just one symbol.
Yes.
But it's true that
Everything is complicated when you. This is another movie where they, because they are they have hematite, which is, has a magnetic interaction, right? And so you can assemble a cluster. What's happening? You can assemble a cluster, and then you can apply an in-plane magnetic field, right? And then then you can drive these clusters around with a magnetic field, right? And so, so this system, is tunable, right? And so this is an example of another, that maybe illustrates a little bit about what is the mechanism at play. Is what they have here is they have just a small hematite particle that's stuck to the interface, okay? And so this hematite particle is, when you turn on the light, it creates, drives the chemical reaction, and that induces flow of the particles towards the source, right? And so that's what happens in this movie over here, right? Is you have the particle, you turn the light on, and you can see that the particles get sucked into the center of chemical reaction. So that's like a sink that creates a gradient either in temperature or in the chemical, and that sink sucks up all the molecules towards its center, right?
And the force here is just the gradient in the osmotic
Huh?
pressure. The force here is on the particles is just the gradient.
I mean that's one possibility. The other possibility is that there's temperature gradients, but I think they argue that it's really the osmotic, the gradients in the osmotic pressure, and so so you can measure. You can measure the velocity of these particles as they're getting sucked into the center, right? And so this is the velocity, right? So this is a particle that's 6 microns from the center, and it's going to move very slowly. And then as it gets sucked into the center, you can see that the velocity speeds up, and that's described as a 1/r² potential, which is consistent with a gradient in osmotic pressure, right? And so this is the nice thing about the system, right? Is it's tunable, right? And so this is the maximum velocity that actually depends on the Debye screening length, but you can see that these things move fast, up to 15 microns per second, right? And then what you can do is you can tune the hydrogen peroxide concentration below the saturation threshold, and you can tune the velocity, and it follows this kind of Michaelis-Menten kinetics that's characteristic of biological enzymes and things like that. And so it's a nice system in that way, is that you can tune its swimming velocity. That's something that it's more challenging to do if you, for example, have a bacteria and you want to speed the bacteria twice as fast or slow it down. That's more challenging.
So you can tune the velocity by changing the intensity?
You tune the velocity by changing the concentration of the hydrogen peroxide, right? So that's the fuel, right? And so by just changing the concentration, you can tune it up or down. Yeah, I mean,
To an ignorant theorist, that's easier.
I'm sure. I mean, yes. I don't know what kind of dependence it is, right? I mean, I think again that's complicated chemistry, but I'm sure it's doable. And so actually, so there's a number of people who have worked on similar systems from a theoretical or computer simulation perspective, and this is just a partial list of all the people that my theoretical neighbors upstairs gave me. And so I just want to show a little bit more, expand on what Christina has told, because I went to my colleague upstairs, Mike Hagan, who's done some simulations on these things with Aparna. And so, so this, I'm just going to expand a little bit on what Christina said and what's going on. Why do these things cluster, right? And so their model is very similar to Christina's model. I think it's essentially the same, right? You have this particle, and it's moving around actively with a certain velocity Vp. And there's 2 major parameters, right? One is, of course, a density of the particles. And the other one, what they define, Mike Hagan, is a Peclet number, right? And so this Peclet number is essentially a ratio of how fast the particle is moving divided by the rotational diffusion constant, right? And so, that tells you, if this Peclet number is very large, that means that I'm just a particle moving along the straight path. And if the Peclet number is very small, or 0, then I'm just an equilibrium system. And so, so this is for one. I think this is for one density, and so this is.
So at the peak there is the translation?
No, that's the rotational diffusion constant. I'm pretty sure it should be, no?
No, it's not.
Oh, it's not?
No, that's
Did I VP is the velocity, sigma is the diameter. That's the diffusion constant. Okay. But
well, it's not. It is. It's V naught squared divided
yeah. Okay. Yeah, it's
what I call the circle, but the
say that again. So, yeah.
it's defined as
Yes.
V naught squared
Yeah.
divided by the diffusion.
Yes. Okay.
So that the whole expression
Sigma should be in the denominator.
is minus sigma dr.
Yeah. Okay.
So it has to be
Say that again. I can.
So if d is d naught squared over d rotational, then the Peckley number is actually inverse.
I was reading this last night at like 2:00, but obviously. Did I?
Divided by d.
So this sigma is the diameter. It's just the cut. I don't know. I copied this last night. I thought I saw it.
Divided by d times c. Divided by d rotation.
Yeah. Okay.
That should be the expression.
Okay. I mean Yeah. Okay. I mean, there's a Peclet number. It tells you if you are. I don't know what its exact is. I mean, yeah, sure. Okay.
Persistence length.
Yeah, yes, it's a persistence length. This is the persistence length over the characteristic length scale of the particle, right? Yeah, okay. Anyway, so you can run these movies over here, right? And then you can see that that they don't run. Okay, it's all right. So let me go. Huh? I know. And now I have the spinning circle.
What's the interaction between different atoms?
It's just hard. It's a short-range repulsive interaction. So it's a VCA potential, but it's essentially like a hardcore repulsive interaction. So there is a repulsive interaction. If you don't have repulsive interaction, this will not happen. This was done by colleagues of mine up one floor at Brandeis, right? Not here, but at Brandeis, by Mike Hagan and Aparna, right? All right, so yeah, no. Sorry, so yes, I have to stop the presentation and exit PowerPoint. That's what at least it tells me. And I'm just doing that because I want to show you this movie, because I think this nicely summarizes. Huh? Is it? Do I need to? Wait, okay. Yeah. Oh no. All right, I know what to do. I prepared for this. This is. Here it is. All right. Anyways, this movie I like, so that's why I'm showing it. And so this is like the entire phase diagram, right? This is density, right? So this is, I'm increasing the density, zero density, high density. Here I'm increasing the Peclet number, right? Or the activity of the particles, right? And so you can see here at very dilute system, as I increase the Peclet number, nothing much happens, right? And if I increase the density, at some point this is gonna crystallize. These, by the way, are monodispersed particles. So they do form crystallites, unlike Christina's model, which was polydispersed. And so what you can see is, as you increase the density, and then at some point, as you increase the Peclet number, you get these large density fluctuations that are shown over here. And then very soon you get into this phase separated regime that is very ubiquitous, okay? And so this thing runs for a long time, right? But now it's equilibrated, and it shows, kind of nicely summarizes in one paragraph, in one movie, the entire phase diagram. So I can run it again, and you can see the phase separation kinetics, right? You can see these phases of dense aggregates growing. Okay. Yes. Well, I mean, what's going to happen is what's going to vary here is the coexisting densities, right? Between, the high density region and a low density region. And so it's very analogous to a gas liquid phase separation. And so I can show you a phase diagram. What do I do now? Go back to PowerPoint. All right. So this is what you see, right? So this is the Peclet number. This is the phase packing fraction, right? And so it's 0 Peclet number. You have uniform density. And as you start increasing the Peclet number, you can see that the density of the particle increases until it reaches a certain point, right? And so here, the density is pretty flat, right? This is P of rho, right? This is the local density fluctuations. So it's very flat until you reach this point over here where it becomes very broad, right? So this is telling you that the density in your system fluctuates quite a bit. So it's very reminiscent of a phase diagram. And then if you go into this two-phase region, you see regions which have a very low density and the regions which have a very high density, right? So in a certain way, it's very analogous to a gas-liquid phase separation and equilibrium system, only that what's controlling the parameter is really the dynamics. And the higher the dynamics you have, the more likely the system is to phase separate, which is kind of unintuitive, right? Because you would think that activity would destroy any kind of phase separation. Does that make sense? And and I guess presumably there's some non-equilibrium critical points sitting somewhere here, but I don't know if people have studied that in any detail. No, so I already stopped that. Okay, and so there's a very simple way to actually understand this, right? So this is your volume fraction, this is your propulsion velocity, and here you have, so what happens is when you have a cluster of particles, right? And so this particle is, going to come to the cluster at a certain rate k on, that depends on the density of how many particles there are out there and at what velocities they are moving, right? And so these particles are going to attach to the cluster at a certain rate, and then they're going to leave the cluster, and that rate at which they leave the cluster depends on the rotational diffusion, right? Is this particle it wants to push in this direction, and for it to leave the cluster, it has to rotate a certain amount and then leave the cluster, right? And so that tells you what is k off, right? And so k off just depends on the rotational density distribution and some fit parameter. And so you can see what is the fraction of the system in the clusters in theory and simulations. And by, the system is in equilibrium when k on is equal to k off, right? When the rate of particles that are attaching to the clusters is the same as the rate of the rate at which the particles are leaving the cluster, okay? And so you have this very simple theory that you can put together, and that fits fairly well with the experiments. Okay. So so that's all I have to say about the simplest, I guess, active matter system. You have isotropic interactions and polar active liquids, and so the real characteristic of this is dynamical clustering, a phase separation of even purely repulsive interactions. Okay. Are there any questions about this part? Yes.
What's the power code?
the color code here. It's the fraction of the particles in clusters, right? So here you have, I guess this is 0, right? So you have 0 particles in clusters. And here you have a very high fraction of the particles in the clusters. All right, so the next thing I want to talk about. So I just spoke for 4 minutes, it says. No, how much? Oh, it's 2:45. So do we need to take a break? Yeah. So I'm really going slow. Okay, I will. I did this. Yes.
First slide.
Oh,
Because you're talking about equilibrium system,
yes,
and then you show the 2D system
Yes.
in ordered phase.
Yes.
XY model.
yes. I mean, it's quasi-U.
I think so the order you showed that does not exist. That's Merman-Wagner.
Yeah, it's quasi-long-range order. Yes, sorry.
No, I was just confused. I thought.
No. I mean,
Okay, good.
I mean,
Just to.
you know,
No,
yeah,
it's fine, it's
the
fine.
order
If you-
doesn't exist, but you know, experimentalist experimentalist is always very difficult for me to check.
No. I'm just
Yeah,
wanna make sure that you-
No, I mean, I know that much.
No, it wasn't like.
Yeah.
I just wanted to check.
Yeah. So who do you work with?
Yeah, I'm
LMU?
making theory in Munich. Germany.
With Irvin Fry, or
Exactly.
what do you work on? Or
So I'm working on biological physics,
Okay.
theoretical biological physics. Actually, I'm a little bit into these bacteria systems and stuff.
okay, like the dynamics clustering, or
Yeah, more population dynamics.
population dynamics?
But of course, that's kind of next door. and
Yeah,
related to
sure.
the- All right.
So my idea is
Not only recorded, also simultaneously webcast.
Oh, really?
No, it's fine. It's good.
Were you there when these guys were working on this, or did you already leave?
No, that was sort of like I actually worked with a little bit with Stefan on the synthesis of those particles.
Oh, really? Okay.
Well, not so much on the synthesis, but more on the. We were playing around with like making patchy magnetic particles.
Mm,
So
mm.
we put one of these cubes
Yeah,
in, and then we
okay.
etched it, and then you get two little Mickey Mouse
Okay, yeah, I
Imitate
think
that,
I
so-
saw that. Or
stuff like that. But, you know, Stefanos,
But
the synthetic genius.
so, do you know
was a
how he's
spectator.
doing? Or
I think he's doing okay.
yeah,
I
he's
think,
been there for a year and a half or something.
No, a little more than that.
Okay, has it already been?
Yeah. I think he's, he's having to find his feet in the chemistry department, which is a different beast. But he's, I mean,
That's what he has to do, right?
he's
No,
gonna-
it's hard
He'll
in-
do fine in the end, I'm sure.
Yeah.
I think it's. I think chemistry is a little funny in terms of politics and stuff like that.
At NYU in particular, or in general?
I don't know. in
I mean,
general,
they
but
don't
I
do
mean-
colloids in US almost at all, right?
Yeah, exactly. So I think he's like one of the only people who does something a little different in the department. And
Yeah.
he's not, doesn't have a bunch of people to discuss with. So
Do you keep in touch with him, or?
yeah. Not in the last 2 or 3 months, but yeah.
Are you
I go to
doing
visit
some
him in New York.
colloid stuff, or?
Yeah, but
Not
not
so much.
so spectacular. At least not in the sense of interesting from a colloidal point of view,
Yeah,
like
okay.
some pattern formation and stuff
Yeah,
like
okay,
that. But
okay.
I don't know. Thinking about.
We've been doing a lot on. I mean, I've started reading about vortices and stuff.
Okay.
So,
For what system?
well, I mean, just in our active mixtures, we see
okay, cool.
vortices now. Well, I mean, I don't know. I mean, in 2D.
Okay.
But anyways, I just have to.
I always wanted
Yeah.
to look at vortices and look at crystals in 3D.
Yeah, that's more
But it's
challenging.
Really
right?
hard to get a
Yeah.
liter of stuff.
Yeah. Oh, you mean just have a? How much does it cost, or?
Too much.
Yeah.
I think we need a friend that has some sitting
Yeah,
around.
okay.
And we can borrow it and give it back. Something like that.
How much do you need, or?
I don't know exactly because I haven't really gotten my hands on it. Depends how viscous it is. I think a liter. Half
Do you need
liter.
a? Do you need a? I mean, with the Lyotropics, you could do it probably,
Probably.
because a lot of these things will form at 1% or something like that.
So it's actually turned out to not because one of the things
Any developments?
No, nothing yet. I mean, I'm just waiting. I mean, I don't have a concrete offers anywhere, so. All right. So let's continue to next model system. So number 2, right? Polar interactions and polar active dynamics, right? And so this is what John Toner spoke about. And so this is the classical Vishek model, right? And what always, kind of intrigues me is that this is so simple, right? I mean, I think one could code this up in a day or 2, right? I mean, not even. And 10 minutes probably, right? So I mean, you don't need something complicated to have an impact, right? And so you have a particle moving at velocity with certain momentum I. You average over a certain radius, and your new momentum is the average all of these particles within certain radius r, and there is you add some noise to it, right? And this is the classical Vishek model. And I just read this abstract, and I thought it's a well-written abstract, right? Because it just summarizes everything. This is how one should write an abstract, right? A simple novel is introduced in order to investigate the emergence of self-ordered motion. In our model, particles are driven with a constant absolute velocity, and then each time assume the average direction of motion of the particles in the neighborhood with some random perturbation added. We present evidence that there's a phase transition, right? And the phase transition is continuous. Anyway, so that's the Vishek model, and again, I guess some of you guys could simulate this in minutes, right, or hours, I don't know. And so what you see is you see observation of these flocks, right? Now, what's when you simulate this, you usually have open boundary conditions, right? And experimentally, that's kind of challenging to achieve, and so that can influence things. I guess you could have things on a sphere or something. So the question is experiments, right? There's this prediction. It's a very simple, elegant prediction, and so what do we do about experiments, right? And I mean, it claims to have relevance to all of these complicated, dynamics of moving beasts and things like that. And so so again, this is the prediction is, as you increase the activity or change the density, you'll have a disordered phase, and then you'll have an ordered phase with increasing density. And so, and the claim is, you have something as complicated as a flock of birds, and this simple theory describes this kind of, very complicated flock of birds. But, how do you really prove that, something like that, right? How do you do controlled experiments on this? And so that's where, so there was, again, a very simple experiment that was developed in the group of Andreas Bausch. And again, it's really very simple, and it's based on a motility assay, right? And so what they do is they run a motility assay, and so the main building blocks of this motility assay are actin motor that is shown over here. Myosin motor and an actin filament, right? And so a myosin motor is really a nanometer size motor that hydrolyzes ATP and takes 5 nanometer step, okay? And so each time it hydrolyzes ATP, it takes 5 nanometer step along this actin backbone. And, that is the building block of something like your muscle, right? And so here's an animation of how that happens. The ATP is released, a new ATP binds, ADP is released, an ATP molecule binds to the thing, to the motor. It hydrolyzes it, and then the thing is released and makes one step, okay? And, Jennifer Ross has studied that much more than I have. And so, this is obviously a very important process, right? Because it really drives the cell out of equilibrium, and it powers a lot of different kind of cellular functionalities that are essential for your life, right? Like muscle contraction, the cell division and things like that. And so what people, in 1980s, early 1980s, managed to reconstruct this motility in a highly simplified in vitro system. And what they do is you have a cover glass, and you cover the surface of a cover glass with the motor proteins, right? And this motor proteins hopefully stick by the tail at the bottom, and then they have their heads. It's kind of funny because motor proteins walk by their heads. They stick out, right? And then what you do is you introduce actin filaments. The actin filaments land on this carpet of myosin, right? And then what happens is the myosin literally, grabs onto actin filament and steps forward, right? And propels the actin filament in one direction. And actin filament, unlike, it has a polarity, so it has a barbed end and a barbed end and a pointed end. I really didn't get much sleep last night. And it goes, depending on the nature of the filament, it's either propelled towards barbed end or pointed end. And so this is a movie of a gliding assay. So what you see here is you see motors sitting below. You have a motor sitting on the cover glass. You don't see the motors. And you have these filaments that land on the cover glass. And you observe this motility as they move forward. And so this is what you see, right? And this is actually, again, I surfed the internet to find a decent movie. This is the best I could find. But this is actually a very bad motility assay movie, right? As you can see, for example, this guy is, it has some motility, but you can see that a lot of these guys are actually stuck. For example, this filament is stuck at its head and it cannot move forward. And so its tail is just beating back and forth because it's buckled by the internal motility, by the propulsion force. Okay, and so the reason for that is that whenever you purify biochemicals, a certain fraction of them are going to be dead. And so here you have some dead motors, and when a dead motor, irreversibly binds, an actin filament. That's also what happens. When you die, literally, you enter a state of rigor mortis in which all of your actin filaments are permanently cross-linked, right? But so that's what's happening in this movie, right? And so, that's the challenge when you work with biochemicals is you really need to obtain very pure samples. So people have been running these motility assays in dozens of groups for dozens of years, okay? Same way that chemists are, propelling their particles for, with different, chemistries. And nobody's ever systematically asked the question, what happens, if I increase the density of the particles, right? And again, it's not like it doesn't take a genius to figure this out, right? But one just has to do it, right? And so that's what the group of Andreas Bausch did at the Technical University in Munich. Why?
Well, you just dissed him.
Oh, no, but I mean, it is a genius, but it's in a certain way because it's so simple, right? I mean,
He's also a really big guy.
I know, I can run faster than him, though. Maybe not in Boulder, though. No, I mean, I can offend him. I don't worry about that. Anyway, so that's what they did, right? And so they increased the density of the filament, and this is what they find. And so so this is obviously a polar particle, right? Unless it gets stuck like this. So you need to make very good care to eliminate all of the dead motors, and there's biochemical procedures that can do that. But this is a polar particle. What the interactions between the particles is not so clear, right? Do they, you could imagine that these things would be nematic or polar. But anyways, this is what they see, right? Now this is really zoomed out. So this is probably 10 microns. but you you go to a much larger field of view. What you have here is labeled 1 out of 200 particles, right? So most of the actin filaments you can't see, and this is what they observed, right? And so you observe clustering, right? Very much like what Vishek predicts, right? You have this dilute phase in the background where you have isotropic actin filaments zooming in all directions, and then you have this cluster of millions of actin filaments roaming around, right? And it's very robust, and it lives for minutes, and so on, right? Okay, and so this is one. I would argue that this is perhaps one of the simplest explanations of the experimental sample that kind of falls in the same category as the Vishek model. Yes.
Sorry, I missed what interaction.
It's actually very weak, right? So these filaments sit on a two-dimensional surface. They move around. They will occasionally bump into each other. As far as I understand, in 90% of the times they pass through each other, but in few percent of the time they'll move in the same direction, right? Because they hit each other and then they reorient. And so it's just like Vishek model, right? You move, you glide on the surface, you move, sometimes you bump into your neighbor and you orient in the same direction, right? Now it's not obvious to me whether that would be a polar interaction. You could also imagine being it anti-polar interaction, right? And I don't know, maybe Jean-François knows better than I do. What they haven't done so far is they and I think they're working on this, they're literally looking at a pair of filaments and classifying the statistics at a microscopic scale, right? And when they encounter each other, at what angle, what happens, right? So those are the kind of Vishek rules that govern the behavior of the system. But it's still, it's interesting because now I can tune everything. I can change ATP concentration. I can change, all kinds of other parameters, and I can, in principle, compare it to the theory. It's easier. I can do this in the lab. I have no idea what time it is, and that thing is. Oh, there is time, 3:07. Okay. If they increase the concentration 5 times, so this previous movie was at 6 filaments per micron squared. If you increase the density to 5 times and a similar labeling ratio, you see something entirely different. You see that you see organization of bands, and these bands propagate in one direction, right? And so here you zoom in and you can really see one of these bands, and it's moving in this direction. Although it's not. Yes.
How does that work experimentally?
Yeah, that's what I was wondering as I was preparing this slide, right? Because at some point it's going to hit the surface, right? And it has to bounce back, right? So, if you have a polar order state, it has to make a circle, or I'm not sure, right? I can Skype him and tell you next Wednesday. I don't know. Maybe do you guys know, or
I know they on the image.
Yeah, so there is actually, okay, so I'll talk about that. There is defects in the system, right? So it's not like all of these things are moving in one direction. There is going to be some defect in certain areas. So you will get microscopic bands that travel through the system, but I don't think it's the entire system, right? So you can imagine this thing turning out somewhere else. Yes. In this movie, or? Okay, there's always schmutz, right? These are experiments, right? Like this stuff? Yeah.
If it's not rebounding off of the schmutz,
yeah, but this is probably just few actin filaments that you can just pass by. But if I if there's few people here, I can pass by. But if I hit that wall,
I couldn't tell. It looked like you could easily have.
Yeah. No, So say that again. Yeah. Yeah. Yeah, I don't know. I mean, I know that they are doing microscopic dynamics like that.
Experimentally, is there anything special about this? It's gonna make more sense.
Okay.
Because I can't recapitulate this. So, what's special about this that is different?
Between microtubules, yeah, you can do exactly the same symmetry of the system, right? Instead of taking actin and myosin, you can take microtubules and kinesin, which is just, huh?
Except kinesians are possessive, and so
That's what I
not
think
the
the
same
difference
way.
is. I think there's a biological difference.
Yeah, but why would that matter? I mean, what only matters is the collisions between the particles.
No, it's not.
No, it's not so bad. Even we can do it.
I do it all the time, but it's not that. I think it's the coordination between the motors. These motors work in teams in biology.
Yes,
Kinesis don't. They work as one team. That's it. They don't work as
yes.
You would think that making the coordination would have to make them multiply.
Yeah, I mean, if I was to sit there and say which system this would work in, I would bet my money it would work in kinesin, but I'm always wrong, right? So, so that when I take that into account, then it's
So, like, when you this, like, with acid,
yeah, but things get very complicated when you have bundles because it's not longer, you have attractions, and you can't. Anyways, I mean, we should discuss this, but I mean, I don't. These are Anyways, I mean, it can emerge, but it does not necessarily have to emerge in a simple system, right? And so this is the phase diagram. This is the density below 5.5 filaments per micron square. You have a disordered phase where each filament goes on its own. As you hit this threshold, you get a formation of these clusters, and over a certain range you have these clusters. These clusters move collectively. Here you can see one of these clusters as it's moving around, and then you increase the density even more, you'll get the persistence length of these clusters increases and their size, and eventually you get these density waves, right? And so here you really have long range direction. And so that's an experimental model system. This was yes.
So, I mean, on the other hand, these kind of density waves, they also, you're calling this a Dirac model. Now,
Yeah, I don't know. Maybe.
what I would want to talk
Okay.
about,
Okay.
because if you have a system where you really have, say, rod-like particles,
Yeah.
yeah.
Yes. Yes.
But they are self-propelled, as is that the role of the underlying motor is to make these
Yes.
filaments actually self-propelled. Then you would get this. And that, so it's difficult to check where it's really for because here the line is not comfortable
Okay. So,
they are considered two different classes.
because you're saying in one case it's the steric effect
Yes.
that and
you
they
can actually show that
okay.
the case where the line is very
Yes.
actually has this
Yes. Okay.
although they would go
Okay. Okay.
in the other case.
Okay. Okay.
So there's still a lot,
Okay. No, I mean, this is,
but you're right, it's very hard to tell which one this is.
yeah. And, they speculate in the paper that hydrodynamics play some role, but it's hard for me to see how hydrodynamics would be important for these things on a two-dimensional surface.
Well, but
Or,
it's a two-dimensional surface with a multi-fluid model.
yeah.
And it's known that when you have a two-dimensional thing interacting with the bulk system,
Yeah.
you can leave the bulk interactions and change the behavior.
Yeah, I mean, yeah.
Actually, I've looked at this.
Okay.
The claim, this isn't actually a
Yeah.
because of those interactions. You actually have to do
You could, I mean, you could actually vary that height experimentally. That's something that I don't, right? I mean, you could make a chamber 1 micron and then everything would be cut off at 1 micron, or you could make it 100 microns.
Even then, there's still complications that come from the fact that there's microtubules who could drag the fluid along with them.
Yes, yes,
The fluid can drag it along with the vessel.
yes, yes, yes,
That creates effective
yes.
forces that also change the dynamics. So it's when I first saw this experiment, I thought it was a great, perfect visualization of the VSEC model.
Okay,
The more I thought about it, the more I talked to people who thought about it more, the more I
Okay.
realized that's actually not true.
all right.
It's very interesting.
Yes,
It's not
yes,
what we
yes,
thought we
yes.
thought it was.
Yes, okay. I mean, so again, when you do, life is complicated. Experiments are complicated. Okay, let me go on, right? And so I just want to give you one quick other example that I think is a great example. And again, it has certain similarities and certain differences. Is a recent paper from Denis Bartolo group in Lyon, right? And so he's made the synthetic model systems of active polar rollers. The previous model I would say is a biosynthetic system, right? You really take individual components and put them together. This is a purely synthetic system, and again, it was discovered in 1896 by Quincke, right? And so if Mr. Quincke just put 50 beads instead of 1 bead, maybe we would have discovered Vishek maybe 100 years ago, right? I mean, it was, again, the idea is the thing, right? It's not like when you do atomic physics where you have to build this heroic setups, right? You just have to have the right idea.
Although the data analysis would have been a lot harder in 1880, whatever.
Right. I mean, I even have colleagues, right, that saw all kinds of interesting stuff in 1970s, and they had, Bob Meyer, he had a PhD student, and this whole, his whole job was to record movies on what they were seeing with a camera. That was like, a PhD thesis or something like that. And so he would have drawn pictures, I guess. Or anyway, so this is a simple system, right? And so it's a colloidal particle that you put in a background, again, dielectric, and you impose an electric field, and of course you get polarization that's that's driven by the surface charges that goes in the opposite direction of this electric field. And what happens is, as you increase the electric field above certain critical point, the system becomes unstable and these things start rolling around. And so it's a simple experimental system. You have a microfluidic chamber. You have these beads that are 5 microns in size. You have thickness of 200 microns. So it's again not 2D but quasi 2D system, or there's fluid definitely around. And so these things, you apply an electric field of about 2 volts per micron, and you have and you have to have a lot of salt in your solution and hexadecanoil. I think this has to have a higher dielectric constant. And what happens is these things start rolling around, right? And so this is one of these Quincke particles, and here you can see a time sequence. They're moving around in this direction. Here they're moving around in this direction. So they have a persistence. They're moving in one direction, although there's some noise, right? And so they have a very constant velocity. This is the velocity probability distribution in the x and y direction. And you can see that it's very constant, but they're equally likely to move in all directions. So this is very different from just taking a magnetic, this is still an active system because you're not imposing a directionality by using an external field, right? These things can move in any direction. You're feeding the system energy from an external field, but you're not imposing any directionality on them, right? And this is what they look like. Okay, so this is important, right? It's a synthetic system, so it's fully tunable. So you have the electric field, and here you have the velocity, and you can see that the velocity drops down, and below certain critical threshold, things don't move anymore. Okay, and so these are these particles, and here is how they move around, right? I don't know, can people see this, or the individual beads, right? They're really zooming, they're super fast. This is millimeters per second, right? So they move a millimeter per second. So, they put to shame everything that biology has invented, right? But, and so this is a dilute system of these particles, actively diffusing in a two-dimensional chamber, right? And so again, obviously, it's a polar dynamics, right? These particles move in one direction. It's not that they go back and forth. And so what happens as you increase the density, right? And so this is probably maybe Mr. Quincke saw something like this. But if you increase the density, okay, it's isotropic, but there is certain diffusion, right? So this is the velocity correlation function. And so this tells you, right, at 0 time, the thing is moves in the same direction, but as you move along time or space, these things become decorrelated, and that's due to the rotational motion of these particles. And so you take a bunch of these rollers, and these are these traces, and you put them in a channel, in an annulus. An annulus is particularly convenient because it's an open boundary condition, and this is what you see, right? Did you see they were initially moving in all kinds of directions, and very quickly they self-organize into this band that travels around along the periphery of the sample. And so here you can see that again. You see these kind of bands, there's many of them, and then they coalesce with one another, and eventually you choose one direction, and you have one microscopic band that propagates along the whole perimeter of the circle. Again, these are not my experiments, but I looked at that and I saw, it could be that it's a instability of some kind, or it could be just an imaging artifact. I don't know how quickly they were imaging it. Yeah, but if you actually move, so I can show you a movie that so this is a different kind of racetrack, and so here you move along here, right? So there's boundary conditions there. Yeah. Maybe that has to do something with it. I haven't thought about this, extensively at all, right? But so again, this is another example of something that looks, like a flock of animals, except that it's a flock of moving colloids. And so this seems like a great model system in that it's again fully tunable, right? The dynamics is obviously polar, but what's not obvious is what are the interactions again, right? So when two of these particles come together, they, there's going to be hydrodynamic interactions, there's going to be electrostatic interactions, and so that's something that Danny Bartolo studied in fair amount of detail, and I think Vincenzo must know more about this than I do because he's worked on it as well. Eventually, it turns out that the coupling between these particles is polar, right? Am I correct? Right. So you have two particles, they come together, they want to move in the same direction. The difference between this system and, for example, the system of Andreas Bausch is that there, with increasing concentration, he saw very well-defined bands. Here, Danny doesn't see any bands, right? He sees one band that moves around the circle, and the size of this band increases as you increase the size of the particles. And they've made larger racetracks, and they always see only one band. So there isn't a striped state that exists, and I think that's what people still are actively investigating, right? And so this is, for example, an example of where I'm increasing the volume fraction. This is, the portion of the racetrack that's occupied, the density of the particles along the racetrack. And so this is the density, this is the position along the racetrack. This is the beginning of the band, and this is the density of the particles, right? And so you can see that the density increases very quickly at the front of the band, and then it slowly decreases, as is shown over here. And if I increase the volume fraction, the size of this band increases, but there's no multiple bands, right? So the more particles I add, the larger the band becomes. You're just resting your head? Oh, okay. Are there any questions about this experiment, or? And so this is the phase diagram, right? Is you have the volume fraction of your beads. Below certain critical volume fraction, there is no organized state. If you've passed this volume fraction, a certain fraction of your system goes into this moving band, and the length of this band compared to the size of your system increases until you eventually get something where all the particles are moving in one direction synchronously, right? And so your whole system becomes a polar band. And so you can see they've taken data for different sizes of racetracks, and that doesn't matter. The only thing that matters is the fraction of the particles that are in your band as compared to the largest to the racetrack size. And so this is at fully packed system. You really have a polar liquid or a polar gas phase. You can see these things pass along and they organize into this state. So this just recapitulates what I said, and so so I guess this is maybe a good place to stop, right? It's 3:30, so I've talked to you about two systems that I actually don't work on actively. One is the isotropic interactions and polar active nematics, and the other system I talked to you about is polar interactions and polar active dynamics. This exhibits this clustering. Polar interactions and polar active dynamics leads to flocking transition that's very ubiquitous. And so what I want to do next time is I want to talk about nematics, right? So these are systems in which you don't have arrows, right? So you have to describe a system with a tensor order parameter. And so there's a couple of realization of these. There's, active nematics of complex living organisms and simple shaken granular rods. And then I'll talk most of the time about our system and compare it to these other systems next time. And then on Friday, I'll just really show you results that we don't understand. And that has mainly to do with, when you take these active systems and you confine them into various kinds of geometries, you see, again, very interesting dynamical patterns.
So you started with a theory.
I did, okay.
Yeah, and now you have all these experiments. So how does this experiment compare with the theory?
Well, I mean, certain predictions, work out pretty well, while others, the robust features of the transitions are can be explained.